Hi everyone, Lauren here. In this video, we're going to go through the EdXLA for the Salters-Nuffield specification, and we're going to look at the A-level biology pre-release article, and I've written some questions to practice that are going to be linked to the topics in the paragraphs of this article that could be similar to the questions that could come up in question 8 or the last question on paper 3. It's roughly about 30 marks worth of questions, so it's a lot of questions, and because it is paper 3, the idea behind these is that they are synoptic, so they cover a broad range of topics from the entire specification. So each paragraph in the article, I've tried to kind of think about things mentioned in that article, questions that could be linked to those topics that are mentioned in the article, to try and make sure that we can have a go at practicing recalling some of that information, some classic question styles that could come up. Now, obviously the article is about hibernation. Make sure you've got your pre-release article in front of you to help with these questions. I'll just refer to the paragraph in each question, just like they do in the exam. So, as typically starts with question 8, it says use the information from the scientific article and your knowledge to answer the following questions. The key bit here is using your own knowledge. What that means is, as we said, there'll be statements or sentences in the article and the questions will be related to them. Doesn't mean there'll be actual answers in the article to help you. It's not like that kind of comprehension. You need to be able to tweak and use your own knowledge, link that to be able to answer these questions, and they're just going to be related to the paragraphs in the article. So first of all, explain how molecular phylogeny would have been used to create the evolutionary tree of mammals in paragraph 2. So they mention this idea of a tree of mammals in the paragraph. That's what I've pulled out as the kind of quote here. So molecular phylogeny, we should be aware of that and what that means. So it means that we're looking at differences in the DNA or the proteins between organisms, and we're using that to create these evolutionary relationships or evolutionary histories, how closely related the organisms are to each other. And then we can use that to build these evolutionary trees or phylogenetic trees that you might have seen in the exam. So this is a 3-mark question, but we don't have to write a lot to get 3 marks here. So comparing differences in DNA between different mammal species to show their evolutionary relationships. So we have to be specific about making sure we're talking about differences, and then we can talk about either DNA or proteins, but that's where the molecule part comes in. And we have to link it to this idea of evolutionary relationships, evolutionary history, or relatedness between species. And we've put different species here, so the idea between differences between individuals or organisms or species, and that's how we build these trees. So those 3 things are what we're going to get the 3 marks for in this answer. Torpor involves a reduction in body temperature. Describe how thermoregulatory mechanisms could bring about a decrease in body temperature. Paragraph 6. So there's obviously a lot of talk about decreases in body temperature, decreases in metabolism and metabolic rate in this article because it is about hibernation. So we're going to take this idea of a reduction in body temperature, and we know from part of our homeostasis part of the course, we should know how the body can bring about a decrease in body temperature. And so that's what we're applying our knowledge to here, sort of. It's not application, we're just going to describe. So it's steps in the process of how this mechanisms or the mechanisms that are available to the body can bring about a lowering of the body temperature. So we should know the hypothalamus is involved. We should know we're sending impulses to the sweat glands to try and stop sweating. We're also going to be causing vasodilation, and that's going to cause us to lose more heat, and a decrease in metabolic rate. All of these things, remember, it's just describe, it's not explain. We don't have to explain how each of these mechanisms brings about a decrease in body temperature. We just have to say what the mechanisms are and what happens. So thermoregulatory centre in the hypothalamus is stimulated and sends impulses to the sweat glands to increase production, and vasodilation increases blood flow to the skin. And the metabolic rate also decreases, and that helps us to reduce heat production because metabolic rate includes respiration rate, and respiration produces heat. So we've got all of those points in there, 3 points. The idea is we're going to get marks for talking about the hypothalamus, as we said, the sweat glands, the vasodilation, and the metabolic rate. So we've actually got 4 points here. So if you didn't get metabolic rate, that's fine. Suggest why it is easier for smaller mammals to decrease their body temperature more significantly than larger mammals. Paragraphs 8 to 9. So this was about talking about how some small mammals are able to really rapidly decrease and significantly decrease body temperature, whereas larger animals like bears actually only decrease it by a few degrees. So why is it easier? And this is a suggested question. So this is where we are really applying our knowledge, taking our knowledge from the spec that we know and applying it to a new situation. So when we talk about small versus large animals, that should always ring a bell in our heads, that is to do with surface area to volume ratio. So the difference here between the surface area to volume ratio of a small mammal versus a large mammal, and why that might make it easier to rapidly drop body temperature. It is because heat is lost faster. Okay, they lose heat faster if they are much smaller mammals because of that large surface area to volume ratio. And also their metabolic rate. So their metabolic rate is normally very high if you're a small mammal because you can lose heat so easily. So you're constantly having to work harder to maintain that higher body temperature. Whereas if you drop it and you don't need to have that higher temperature, body temperature, then your metabolic rate can also lower quite significantly because you are a smaller organism with, fewer cells and things like that. So if you're not maintaining the body temperature, that can also help. But even if you didn't know the link between surface area to volume ratio and metabolic rate in a small organism, you could just talk about the fact that the difference is surface area to volume ratio and then talk about the kind of loss of heat. So small organisms have a larger surface area to volume ratio. It's going to get us 1 mark. And this means they lose heat faster as there is a shorter distance for radiation. A 2nd mark. You could have put diffusion if you want to, although heat technically doesn't diffuse, it radiates or conducts, but I think they would accept that idea here because you're applying your knowledge of diffusion here to kind of heat energy. But again, you've got the metabolic rate comment as well if you did end up talking about that, but you would get 2 marks just for talking about the loss of heat because of the larger surface area to volume ratio. Explain what would cause the change in the level of gene products in cells from the same tissue during torpor stages. So this is paragraph 10, where they've clearly taken tissue samples and looked at the gene products being produced over time at different stages of torpor, kind of before, during, after. So we need to give reasons. That's why it's an explain question that could cause. And this is a direct quote from the text, which sometimes you'll see in these questions, but not always, the change in the level of gene products. When we talk about gene products, that means mRNA or proteins. So what could cause the level of mRNA or proteins from genes to change within the cell? And specifically, these are cells from the same tissue. So all these cells are the same. So it's got to be something that might change the gene expression in all of the cells at once in a similar tissue. So it's going to be about gene expression. It's going to be whether they're activated or switched on or switched off, and therefore how much mRNA is produced. If your genes are not activated and expressed, then you don't get mRNA, which means we wouldn't get proteins. So we have to make sure that our explanation is going to lead to a change in whether or not mRNA or proteins is produced. So all cells in the tissue receive the same stimulus, which has to be, during the phase of torpor that they're in. This can then either activate or deactivate gene expression. And if genes are activated or switched on, then mRNA will be produced. So we don't have to go into loads of detail here for 3 marks. We have to show that we understand that all cells in the same tissue, all the cells are identical. They must all receive the same stimulus, external stimulus, about what stage of torpor the animal is in or going into. And then we have to understand that that must affect gene expression. You can probably say either activate or deactivate. It doesn't really matter because it just says the change in the level of gene products. It doesn't specify whether it's increase or decrease. And obviously it's through the different stages, so that will change as we go through. And then we just need to link that to make sure we're showing that we understand when we talk about expression, it's going to change the amount of mRNA that's going to be produced. and that's our kind of showing our knowledge of what we understand by gene products, and that's going to get us the 3 marks. State what is meant by the term genome. You will probably have a state what is meant by the term question for a comprehension question. They normally find a keyword within the text that isn't defined, so it doesn't give you the definition, it's just used in context, and we should be able to define it. The only one I could really think of was genome when I read through it. So there might be some others that I might have missed, but like tissue, for example, you could get a definition of a tissue. But state what is meant by the term is just a definition, so it's used in the paragraph. It's not really going to give you any clues in the paragraph as to what it is necessarily, but you can always have a reread of that paragraph 11, find the term genome, see how it's used, and then see how you're going, think about your answer. But hopefully we should just know the definition of a genome is that it is all or the complete set of genetic information in an organism. Explain why high blood pressure increases the risk of developing cardiovascular disease. Paragraph 13. So this is a question, one of those ones again, that is only slightly related. High blood pressure, coronary heart disease mentioned in the paragraph. So we are just taking that as a link. We should know some of this stuff from obviously topic 1. So reasons why high blood pressure increases the risk of developing coronary heart disease or cardiovascular disease. We should know the links between those two things and why it's obviously a risk factor. So that's what it wants us to explain. It's not really related to the topic because it just sort of talks about the fact that there are potentially molecules produced that can help protect against this. So this is just a kind of linking point to jump off from to get us to show our knowledge of cardiovascular disease. So we know that it causes damage to the artery lining, and then that's going to cause an atheroma formation, which then narrows the arteries and reduces blood flow, which obviously leads to coronary heart disease. So high blood pressure causes damage to the artery endothelium. Very important that you use that correct term there. It's the immune kind of inflammatory response to that damage that then leads to the atheroma formation. And that increased plaque buildup or cholesterol buildup narrows the lumen of the arteries. And that reduces the blood flow. Give 2 current methods for reducing high blood pressure. Again, you've got another jumping off point. So they've talked about high blood pressure. They've talked about the fact that there might be these molecules that might be able to protect against it. And they've talked about in paragraph 14, this idea that maybe using some of these findings to try and design drugs. So although it doesn't specify drugs, we should know that currently being used to try and treat or reduce high blood pressure are antihypertensive drugs. I think you probably would get away with naming 2 specific antihypertensives if you wanted to, as long as they were correct. But I would think it says methods. It doesn't say drugs in the question, so we can also think about some other things we can use. So some lifestyle choices, things like reducing salt intake and stopping smoking to try and reduce high blood pressure. Because if you've got high blood pressure, those could be the cause, as well as something genetic as well that require medication. So there's options here for you to put down. You only need 2, so just pick 2, whether that's 2 different types of hypertensive drugs. And I would say antihypertensives and then name 2 different ones, like beta blockers and something else, for example, or go with some of the lifestyle ones. So taking antihypertensive drugs such as beta blockers and also reducing salt intake in diet is the 2 that I've gone for. Explain why exposure to cosmic radiation could cause mutations. And this is in the box paragraph B, where we start talking about space travel in humans. We should be hopefully aware of some of the things that can cause mutations, and one of those being ionizing radiation. So we need to give the reason why the exposure to the ionizing radiation causes mutations. We can't use the word mutations, but we need to give the link between the exposure to the radiation and what that could cause, why that could cause a mutation. Really, we're saying, do you know what a mutation is? It's a change to the base sequence in the DNA, and how come the radiation can cause that? It can cause that because it can damage the DNA. So damage from radiation of the DNA can lead to a change in the base sequence, which is a mutation. So because ionizing radiation can damage DNA, the damaged DNA gets you the 1 mark, and then causing changes to the base sequence, which gets you the 2nd mark. Okay, so often if a keyword is used in the question and you're giving, and it's an explain question, it's an opportunity for you to show that you understand what that key term means, in this case, mutation. Explain why lowering the body temperature to 32 degrees would decrease the metabolic rate in the body. Box paragraph C. So this is what we're talking about, lowering the human body temperature to try and, or induce this state of hibernation or torpor. So we need reasons why. In this case, we're not kind of commenting on the space travel or anything like that. We're commenting on why that decrease in body temperature would then result in a decrease in metabolic rate. So reasons why, obviously our normal body temperature is 37, so to drop it down to 32 would be a decrease. Why would that then lead to a metabolic rate decrease? Remember, with metabolic rate or metabolism, that is all the chemical reactions in the body. That includes respiration. And all of these chemical reactions are enzyme controlled reactions. So this example, we've had the example about how the body can lower the body temperature, but this example is what would be the impact or the result of lowering the body temperature. It would lower the metabolic rate, and they just want the why. So lowering body temperature, there would be less kinetic energy. Therefore, there's going to be fewer collisions between enzymes and substrates. So the metabolic reactions or chemical reactions, including respiration, would slow down. So this really is an enzyme question. Okay? Anything about chemical reactions and temperature is always going to be about enzymes, and this is no different. So lowering the temperature lowers the kinetic energy of enzyme and substrate molecules. This means there's fewer successful collisions, so fewer enzyme-substrate complexes are made. This slows down the rate of metabolic reactions, including respiration. I would say the main marks here would be for kind of the kinetic energy, the fewer collisions and fewer enzyme-substrate complexes forming, and the idea that you can link that to then slowing down the metabolic reaction rate and link to respiration, for example, I would. That kind of really sums it up at the end and links it back, so you're definitely showing how you understand that those reactions are chemical reactions that need enzymes. But I think the main thing here would be kind of the enzyme part of the answer to get you those marks. This is obviously just an example of a question that could be related to temperature and enzymes. They could give you it in other forms. So for example, in paragraph 16, where it talks about hypothermia, they could ask about why hypothermia leads to metabolic stress. That would be the same kind of answer, because hypothermia, low body temperature, why would that lead to metabolic stress? It would be the same answer in terms of the enzymes and what happens. Or there's another point where they talk about where drugs don't work to treat hypothermia at very low temperatures. Why don't they work? Well, drugs won't work at very low temperatures because, again, less kinetic energy, less chemical reactions occurring in terms of the speed because of fewer successful collisions. So you could get this same style of enzyme answer for lots of different questions throughout this text, because there's lots of mentions of lowering of body temperature or lowering of temperature. So have a go, think about this question and make sure you're happy with this style of answer, but then just be prepared to use that kind of knowledge and apply it maybe to a different type of question to this in a different paragraph where it mentions lowering temperature. The diagram shows the molecular structure of adenosine and caffeine. Caffeine is a competitive antagonist of adenosine. Explain how caffeine activates neurons with adenosine receptors, causing feelings of alertness. Paragraph 15. So, whenever two molecules are shown to you next to each other, often it's to try and demonstrate that their molecular structure is similar. Okay? So we've got two bits, hopefully you can see those, the two bits of adenosine and caffeine that you can see are similar to each other. We know that it says that caffeine is going to activate neurons. That means it must cause an action potential. and it's activating those neurons that have adenosine receptors, so therefore it must be able to bind to those receptors. That also supports this idea that it must have a similar molecular structure. It causes feelings of alertness, and it says that it is an antagonist of adenosine and competes, it's competitive, it competes for the active site or competes for the binding site of these receptors. We know that antagonist means opposite, and we know from the text that the adenosine causes feelings of sleepiness, tiredness, so that caffeine has the opposite effect and causes the feelings of alertness. So if when adenosine is binding you feel sleepy, that means that potentially those neurons are not being activated or they're being inhibited, or fewer action potentials are being released because it's trying to reduce the effect or reduce the stimulation from those neurons. So if we put this all together, this is what it wants us to talk about, but it wants us to specifically explain how it can do that. So it's using your knowledge of how neurotransmitter-like chemicals binding to receptors on neurons, that steps in that process that's going to lead to an action potential. So this is one of those application style questions. We've again taken something very small from the text. We've given you some more context to help you to interpret this new information, but then we are just going to be applying our knowledge of how neurons are activated by things binding to their receptors in order to kind of produce this answer. Okay, so it's 4 marks. If we think about our main points, we have to talk about the fact that caffeine and adenosine, caffeine is a similar shape, okay, because we're going to then need to say that in order to be able to say it binds to the receptors. It doesn't say any of that in the question. It leads us there, but it doesn't say it, so we have to be able to say that because that's the process of how caffeine can do this. It's going to cause an influx of sodium ions because it's going to open sodium ion channels. That's going to lead to depolarization on the postsynaptic neuron, and that's going to then cause an action potential if it is above the threshold. So caffeine has a similar shape to adenosine, so it can bind to receptors. It causes sodium ion channels to open and sodium ions to diffuse into the postsynaptic neuron. This causes depolarization of the postsynaptic neuron, and if a threshold level is reached, an action potential is triggered. Okay, so you've got 4 points there. Talk about the similar shape and it binding, causing the sodium to move in, that leading to depolarization, and then that leading to an action potential. I think there's probably maybe more than 4 marks worth there, but that is the whole story, and I wouldn't want to leave too much of that out and risk losing marks, but as long as you've got the main points there, and especially the first point, because that's the kind of application part of this question, isn't it, about understanding about the similar shape. Discuss the ethical issues relating to the use of rats and pigs in experiments similar to those described in the article. Paragraph 16-17. I actually also think it goes into paragraph 18 as well, where they mention the pigs. But the idea behind this is just because there is mention of use of animals in experiments, we could be asked about these ethical issues. That is a classic question that comes up. So discuss means we kind of need to give the pros and cons. We need to give both sides of the argument here. And kind of the ethical issues are kind of like the harm done to animals versus the usefulness of this research and the benefit to humans. And we're talking about rats and pigs here, so we're not talking about invertebrates that don't have a fully developed nervous system, so they can potentially feel pain, they can potentially feel suffering. With these, it is just best to learn kind of 2 arguments for, 2 arguments against. They don't really change in terms of their answers. You can learn these as kind of statements and then use them depending on the style of question. There's 4 marks, so we need to try and give 2 arguments for and 2 arguments against. So one of the arguments for is always that some experiments, the benefit of the research, for example, to human health, outweighs the potential harm done to any animals. And the other argument is that there are regulations that mean the animals must be treated or kept well or treated humanely as far as possible. You could potentially think about the fact in this example, they're not being given a disease, they've not been bred with any kind of disease, not being given cancer, and they are just being kind of like, you know, put to sleep, put to hibernation, and then woken up again, which isn't something that is as maybe harmful or suffering compared to other studies. Against the use of animals, again, these arguments don't really change. The idea that all animals have rights just as much as humans, or humans don't have any more rights than animals. And the work on animals may not be applicable to humans. So we're doing all this study and research on animals, but we don't know if what exactly will happen and the patterns and things that we see in these animals will translate directly to humans. So it may well be just as useful to use cells and tissues or computer models to do the work instead to prevent the suffering. There is also the other one that animals can't give consent. Unlike humans who can give consent to take part in studies, animals can't. So there's another option there for your against argument. But like I said, most of these arguments are something that we can just learn and we can use to answer these ethical questions when they come up. Describe how energy is produced in the mitochondria through oxidative phosphorylation, paragraph 17. So again, I've taken the mention of the fact that mitochondria help provide energy and generate heat through respiration, although it doesn't say respiration in the question. So I've taken that and we're going to use our knowledge to be able to describe again the steps in the process to show how mitochondria produce this ATP through the specific bit of respiration, which is oxidative phosphorylation. We don't need to give full details about glycolysis or the Krebs cycle or any of the other stages. It's told us to focus just on this question to get the 4 marks. So hopefully we're happy. We're going to be talking about the electron transport chain, how we're going to produce ATP from ADP and inorganic phosphate, the fact that we're going to be using the process of the chemiosmosis with the hydrogen ions, and you could talk about oxygen being the final electron acceptor as well. Although it is necessary because it means it completes the chain, and you could get a question about oxygen as the final electron acceptor, but I think that would be very specific. But we just need to make sure we've got 4 points that accurately describe how ATP is being produced through this process. So electrons are passed along the electron transport chain. You can add the detail that they are from the reduced NAD and FAD if you can remember. As they move from one protein to the next, they lose energy, and this energy is used to actively pump hydrogen ions from the matrix to the intermembrane space. The hydrogen ions then diffuse back through ATP synthase. This is obviously an enzyme that catalyzes the formation of ATP from ADP and inorganic phosphate. So I haven't mentioned about the electron acceptor, final electron acceptor being oxygen. You could as well. I think this is more than enough for 4 marks. If you didn't go into the detail of the chemiosmosis, so the hydrogen ions moving and the concentration gradient being produced, you could talk about and just use the word chemiosmosis as a brief description as well, and that probably will also get you marks. Name another part of the brainstem that is responsible for controlling automatic bodily processes, paragraph 18. So this is talking about part of the brain region that they've looked at, and it's called the raphe pallidus. So this is about asking for another part of the brain that we are aware of that's responsible for controlling automatic bodily processes. It's only 1 mark. It's hoping that you can recall a part of the brain that is responsible for part of this. Automatic bodily processes include things like heart rate, non-conscious control, so hopefully we're happy that that would be the medulla oblongata. There could potentially be other brain structure and function related questions related to this paragraph, so it also might be worth having a think about those. Explain how the hypothalamus can coordinate the raphe pallidus in the brainstem to decrease neural activity. So this is explaining how, kind of the reasons, the steps in how this can happen. And it's about linking this idea about how is the hypothalamus going to be connected to another part of the brain and how will it control the neural activity. And if it's decreasing it, we need to be thinking about the fact that it's going to be fewer impulses, fewer action potentials, And what type of the nervous system we're going to be using? We're going to be using parasympathetic nerves. Okay, so parasympathetic, remember, think para like paralyzed. We are reducing, slowing down, preventing, all of those things where you're kind of slowing down heart rate or slowing down anything like that uses the parasympathetic nervous system. So that's where it wants us to kind of apply our knowledge here. A bit about our knowledge of the hypothalamus, knowledge of how impulses are sent in the brain and how we can slow things down using nervous impulses and the knowledge of the parasympathetic nervous system. So it sends impulses along parasympathetic nerves to decrease the frequency of action potentials to the raphe pallidus. Explain how a change in gene expression could slow down the rate of cell division, paragraph 19. So rate of cell division, we're talking about mitosis obviously here, and there is a thing there about how cell division gets slowed down in this paragraph in 19, and it's linked to this idea of how could you have a change in gene expression. This has come up quite a lot actually, so as well as kind of the metabolic rate and temperature, we also have lots of talk about gene expression, gene control, control of transcription, translation in cells, because all of this is kind of linked to how these changes occur in the body and in cells. So in terms of gene expression, again, we're thinking about proteins being produced. We should hopefully know about the cell cycle, mitosis, that it's controlled, it's regulated by checkpoints, and these checkpoints, are controlled by proteins at each point to kind of check and make sure the cell can progress to the next stage. So our answer needs to be about our understanding that gene expression is going to lead to proteins. Those proteins can have a handle or a control in the rate of cell division by being checkpoints and controlling whether the cell cycle continues and speeding it up and slowing it down. So if the expression of a gene that codes for a protein that is involved in regulating the cell cycle is changed, that could inhibit or stop or prevent cells from passing checkpoints in the cell cycle. This would cause the cell cycle to be lengthened or inhibited by checkpoint proteins, for example, from tumour suppressor genes. So just this knowledge of the cell cycle, the fact that there are checkpoints, the fact that the cell cycle is regulated by proteins at these checkpoints. If we change the gene expression for genes like tumour suppressor genes that are responsible for these proteins, then that's going to affect the cell cycle and how it's controlled. So you could go just one way and say there's a protein, it stops cell division at a checkpoint, or normally if it's not there, cell division continues, but when it's there, it stops it. So if you express that gene and produce that protein, the cell cycle could be stopped or slowed down. So you could go just kind of one specific example. I've kind of said about it changing, could inhibit, could speed up, slow down, okay? But it does say slow down in the question. It doesn't say change. It could, obviously, it's not a complete prediction. You might get something about the cell cycle and cell cycle regulation in a different context, not necessarily just about slowing down, but just know that that could be a link there. Lysosomes are used to break down cell detritus to be recycled or removed from the cell. Explain how lysosomes work to break down and remove debris from the cell. Paragraph 19. So paragraph 19 is talking about one of those kind of the cellular damage that comes as a result of aging, and that actually the pathways and things that can help with reducing the aging break down these detritus and bits that would normally build up or cause cell damage instead. So they have given us the information in this question that it is to do with lysosomes. Okay, we should know what lysosomes are as an organelle. We know that they contain hydrolytic enzymes called lysozymes if you want to, but other enzymes as well. So if we've been told that it's lysosomes that are being used to break down the cell contents, we can link our knowledge here with things like phagocytosis, and it's explaining how they break it down. So that's using our knowledge of the hydrolytic enzymes. And then also, how does it get removed? So we should also hopefully know that lysosomes are vesicles. They can fuse with the cell membrane. Contents can be removed from the cell. So we've got kind of a organelle, cell structure, function question, and it's just being related to kind of maybe a different context to phagocytosis, how you might have learned about lysosomes. So lysosomes fuse with vesicles containing debris, and enzymes hydrolyze the molecules. I've put that in a context here with this idea of the lysosomes fusing with a vesicle that has the debris inside. It doesn't have to be that if you just talked about the fact that the lysosomes contain enzymes that can hydrolyze or break down the molecules. You can't use the word breakdown though because that's in the question, so we would need to talk about hydrolysis. And then the lysosome or the vesicle, depending on which way you've talked about it in your answer, fuses with the cell surface membrane and releases the contents. Again, you can describe or just use the phrase exocytosis to get that second mark. Explain how environmental factors could result in epigenetic control of neuron cell activity. Paragraph 20. So again, we've got like another gene expression question, but this time related to epigenetics, which we haven't had up until this point. And this idea that we know that hopefully we can talk about methylation and acetylation of histones, methylation of DNA, acetylation of histones. But this idea that obviously this whole way through we've been talking about the fact that hibernation happens as a natural process in response to environmental changes. So when it happens during the winter or it happens as a result of cues from the environment, temperature, light intensity, etc., then the idea that presented in paragraph 20 is this idea that we could use these environmental cues to start figuring out how to change that gene expression, obviously without changing the base sequence, which is what epigenetic control is. So again, we've got this idea, we've led you down to the idea of epigenetic control. The environmental cues are going to be cues from the weather, as we said, or the seasons and things like that. It says that in the paragraph. And then we're going to need to mention methylation of DNA or acetylation of histones. Normally you can mention one or the other and still get full marks. I would say if you're going to talk about histones, maybe talk about how the DNA is wrapped around the proteins called histones and then link that to its unwinding to make sure you get the full 3 marks. Or you can mention both because you can. It just says epigenetic control. And again, we're talking about gene expression, so it has to be linked to whether or not genes are expressed in the neurons that's going to lead to changes in cell activity. So acetylation of histones causes DNA to unwind and allows RNA polymerase to bind. Again, you could mention before this about the histone proteins are bound to DNA. DNA methylation can also affect the transcription of genes. And these can change gene expression in neurons. Okay, so we have to mention gene expression. It's not mentioned in the question, it's mentioned in cell activity. You could obviously mention about RNA polymerase binding and then leading to transcription. That's the same as talking about gene expression, okay? Make sure if you're talking about epigenetic changes, you only relate acetylation to histones and you only relate methylation to DNA, okay? You can't mix those up because then they would be incorrect. Okay, those are all my ideas that I've gone through as many of the paragraphs as I think are relevant and found things that link to it. Obviously, there could be some actual other things that I've missed, but like I've said, in places where we've got these general themes, so temperature, metabolic rate, enzymes repeatedly coming up, acetylation of histones and the epigenetic side, but also the other sides of gene expression, so gene expression, mRNA, external signals from cells, all of those things, that comes up as a theme because that is sort of how these changes to the cells and changes to the body are occurring, so that's definitely something in there. Bits about the brain and bits about obviously homeostasis and the temperature control regulation and bits about potentially things like the heart and the cardiac cycle and cardiovascular disease because it's mentioned in the question and some other little things dotted through. So hopefully this is a good preparation and a good set of revision of these topics ready for paper 3.